Dear friends, I welcome you to this lecture session on chromosomal aberrations and chromosomal disorders. What are chromosomal aberrations? In healthy individuals, in normal individuals, the chromosomes will be normal with respect to the number, with respect to the structure. Sometimes what happens, there will be a change in the number of chromosomes, there will be a change in the structure of the chromosomes. So that is a deviation, that is an aberration. So such studies, because of the chromosomal aberrations, chromosomal disorders in human beings, in animals, in plants, in other organisms. So remember, any abnormality in the structure and number of chromosomes is known as chromosomal aberration. Chromosomal aberrations lead to chromosomal disorders in human beings. The chromosomes carry the genetic information composed of DNA on framework of protein. Chromosomes are nucleoproteins. Important part is nucleic acid, DNA. So to make it stable, to make it replicate, to make it very safe, the proteins are present, histone proteins. Segments of DNA molecules comprise the genes. DNA is a very long molecule, a linear molecule. A number of genes are there. A segment, a fragment, a part of the DNA is gene. Genes are the units of heredity. Genes are units of the heredity. During cell division, each chromosome can be seen to consist of two parallel strands, the chromatids. During mitotic division, each chromosome consists of two chromatids during the metaphase, and these chromatids get separated during the anaphase and they transform into the chromosomes, held together at one part known as centrome. Karyotype. What is a karyotype? Suppose an organism, just like a rice, has 24 chromosomes. We are representing the 24 chromosomes in a diagrammatic form with respect to length, with respect to the centromere, with respect to the arms. We are showing them in a diagrammatic form. That is called karyotype. For example, all the 46 chromosomes of human beings are shown in diagrammatic form. That is karyotype. It is a systematized arrangement of the chromosomes of a single cell according to length, or according to centromere position, according to the arms, or according to the telomeres. So it is a systematized arrangement. In the human cell, there are 46 chromosomes, 23 pairs have diploid number. Of these 23 pairs, 22 are similar in both sexes. 22 pairs are similar in all respects. That means with respect to length, with respect to centromere, with respect to the length of the arms, the thickness. Then a pair is known as allosome or sex chromosome. Then these allosomes in female are XX, XX, In male, they are XY. Therefore, in male, 22 plus XY. In female, 22 plus XX. Chromosomes are arranged in groups A to G according to their shape and size. So they are classified according to the shape and size. We will see. So this is a karyotype of normal male. So here you can see 22 chromosomes. Look at here. All of them are not similar. There is a lot of difference. The first chromosome, second, third, like this. There is also a difference in the position of the centromeres. There is also change in the length of the arms. Similarly, telomeres. Can you see? Some are metacentric, some are submetacentric, some are telocentric, some are acrocentric. Like this 22 chromosomes. So here you can see XY. X is different from Y. Y is very small. Y is very small. So 22 chromosomes, these are autosomes, common for male and female. These are grouped A, B, C, D, E, F, G. So in normal male, this karyotype will be like this. 22 plus a pair XY. This is a normal situation. If the chromosomes are like this, then the individual will be, that male individual will be normal. Similarly, you can see here female. X, both are similar. XX are similar. 22 plus XX. So in a normal female, the chromosomes will be, karyotype will be like this. Karyotype. Then idiogram. What is the difference between karyotype and idiogram? Sometimes called karyogram. Suppose if you take a particular chromosome, there will be genes arranged linearly, one above the other, one after the other. If these genes are represented in the form of a band, suppose 12 genes are there, 12 bands. Like that, for every chromosome, if you represent the genes in the form of the bands, that will be known as idiogram. That will be known as idiogram. So, idiogram information will be more useful than the karyotype, because we will be knowing on which chromosome which gene is present, where is it present, and to which gene it is related, which gene it is linked, in which gene it is non-linked, like that. Ideograms are useful in identifying various abnormalities associated with a range of chromosomal disorders and determining the links. So this is, you see, suppose take a particular chromosome. The genes are represented in the form of bands. This is called idiogram. But in karyotype, just we represent a diagrammatic representation, we don't denote or we don't indicate the number of genes, position of the genes on the chromosome. So we will be knowing only the length, width, centromere position, such type of details. Chromosomal aberrations, abnormalities. Aberrations means abnormalities. So you take a human being, male, for example, you take 22 plus XY, female 22 plus XX. This should be the normal. So in all the 22 chromosomes, the structure should be normal. The number is normal. Suppose if there is a slight change in the structure of a single chromosome out of 22 or out of 23, even a small change in one chromosome out of 23 or a small change in the number of chromosomes, structure of chromosomes, or change in the number of genes. Change in the number of some. Originally there are, for example, let us suppose there are 15 genes. Suppose these 15 genes are reduced or now represented only by 12 or 14, one gene less or 3 genes less. Similarly, change in position of the gene, same genes will be present, but the order will be changing. All such abnormalities will lead to change in the, can be lead to aberrations, a drastic change in the phenotypic number, structure, number of genes, arrangement of the or sequence of the genes. Any change in these four, that is considered as a chromosomal aberration. So chromosomal aberrations can be broadly divided into structural, numerical. Number. For example, let us take this first, numerical, means number. So what should be the number? 22 plus XX, 22 pairs plus XX or 22 pairs plus XY. Euploidy, aneuploidy. Euploidy, euploidy. For example, 23, 46, 69, 92, like this. This is called euploidy. 2n, 4n, 6n. Instead of 46 chromosomes, there will be 92 chromosomes, 2n. Or 4n. So change in the complete set of chromosomes. Instead of one set, there will be 2 sets, 3 sets, 4 sets, 5 sets, like that. But in these sets also there is a change. 2n, 4n, 6n. But here 3n, 5n, 7n. Two n, 4n, even number, odd number, 3n, 5 and 7. This is called autoploidy. It is alloploidy. Suppose in individual or you take Drosophila, the 8 chromosomes. 2n means 16, 4n means 32, 6n means 48. Here 3n means 24, 5n 40, like that. Autoploidy, alloploidy. Aneuploidy means change in the 1 or 2, not sets, but 1 or 2 chromosomes, less or more, lesser, not complete set. Suppose 8 chromosomes are there, not sets, but 7 or 9 or 10 or 6, like that. Nullisomic, 2n minus 2. 2 chromosomes will be less. Monosomic, 1 chromosome instead of 46, 45. Trisomic, 2n plus 1. Instead of 46, 47 chromosome extra. That is also problem. That is also that causes some problems. Then tetrasomic 2n plus 2 instead of 46, 48. So all these are numerical changes. Then structural number will be same, 46 chromosomes in man or 8 chromosomes in Drosophila or 24 chromosomes in rice, normal. But in 1 or 2 chromosomes there will be a problem. A part of the chromosome may be deleted, that is called deletion. A small part of the chromosome. Or duplication. A part of the chromosome is duplicated, repeated. Then inversion, change in the sequence. Suppose there should be A, B, C, D. See that A, B, C, D or A, B, D, C. There is a change in the sequence of the genes, that is called inversion. Translocation, exchange between the non-homologous chromosomes. A piece of the chromosome is given to another chromosome, but that is non-homologous. So this is a brief summary of the chromosomal aberrations, or either chromosomal structure, chromosomal number, just now we have seen. In human beings, they are very common and cause early spontaneous miscarriage, abortions. Suppose a fetus is with trisomic, nullisomic or tetrasomic, any abnormality like deletion, inversion, other thing. So that is an aberration. There will be problem. So in such cases, in most of the cases, miscarriages, abortions will occur. But not always, cause multiple congenital abnormalities and learning difficulties. Some are born with defects. If chromosomal aberrations are there, sometimes the babies are born but with some defects. They are called chromosomal disorders. Chromosomal disorders. So chromosomal aberrations or deletions, a part of the one chromosome out of 46 removed, deleted. So that also a problem. A part is repeated, A, B, C, D. For example, A, B, again A, B, C, D. Means A, B part is duplicated. In first case, A, B, C, D should be there, but A, B part will be missing, deleted, only C, D. Inversion, as I told you, A, B, C, D, that is normal, but A, B, D, C. Translocation between non. Numerical aberrations, polyploidy, multiple of the haploids, aneuploidy, abnormal number, individual change in the individual number of chromosomes, either 1 or 2, but here sets, complete set, 2n, 3n, 4n, 5n, like that. Then deletion, loss of a portion of a chromosome, A, B, C, D, E, F. That is the sequence of the genes in a particular chromosome. But a small part lost. Only C, D, E, F will be available. That is duplication, extra piece of chromosome. Inversion, fragmentation of chromosome followed by reconstitution with a section of inverted, translocation, transfer of chromosome or segment of it on a non-homologous chromosome. Deletion, loss of portion of a chromosome, two types, terminal, interstitial. The deletion may occur at the tip. For example, A, B, C, D. AB is lost, terminal part is lost. For example, A, B, C, D, E, F. A, B, E, F. CD part is lost, that is interstitial. Between, middle part. The deletion causing agents. Which agents cause the deletions? Heat, radiation, viruses, chemicals, errors in recombination, result in partial monosomy. Example, Cri-du-chat syndrome in children. Then chromosomal duplications. A, B, C, D, E, F. A, B, C, D, again C, D. CD part is duplicated. 4 types: tandem, reverse, duplication of different arms, non-homologous. Causes: duplications often result from unequal crossing over during the meiosis. The crossing over will be perfect, it should be normal, exchange should be normal. But one will get the other fragment, one will not get the other. Can occur via errors in replication during S phase. You know the S phase, cell cycle. G1 S, G2 M phase. Deletions in humans cause so many diseases, syndromes. All these are because of the deletions, a part of the chromosome. Inversion, change in the order, may be paracentric or pericentric. Change in the order only in one arm of the chromosome. Suppose this is centromere position, this is one arm, this is another arm. Change in the order only in one arm, that is paracentric. Suppose change in the order here also, that is pericentric. Translocation, transfer of a portion of chromosome to another chromosome of non-homologous chromosome. For example, from 12th chromosome, a small part is taken out and inserted on the 19th chromosome. 12th and 19 are non-homologous. Terminal translocation, reciprocal translocation, Robertsonian translocation, the fusion of long arms of acrocentric chromosomes. Acrocentric chromosome, Robertsonian. Variations: polyploidy, aneuploidy. Polyploidy, 2n, 3n, 4n, 5n, 6n. Then in that, even number 2n, 4n, 6n, like that. Odd number 3n, 5n, 7n, like that. Aneuploidy, individual chromosomes, only just 1 or 2 chromosomes, not the sets. Ploidy,